Hello, everyone, and welcome to Akoya Biosciences webinar series. Today's presentation, Validating Multiplexed Antibody Panels for Pancreatic Research Applications, is presented by Dr. Diane Saunders and Dr. Grady Carlson. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more about our sponsor, please visit akoyabio.com. Now, before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or you can use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. Speaking first today, we have Dr. Grady Carlson, Technical Application Scientist, Akoya Biosciences. Following Dr. Carlson, we have Dr. Diane Saunders, Staff Scientist, Powers and Brasova Research Group, Vanderbilt University Medical Center. For complete biographies on our speaker, please visit the Biography tab at the top of the screen. Dr. Carlson, you may now begin your presentation.
Thank you for the introduction. My name is Grady Carlson, and I am a Technical Application Scientist for Akoya Biosciences. Today, I'm going to be talking about the CODEX solution, which allows for highly multiplexed tissue profiling for single-cell spatial analysis. The need for multi-marker spatial analysis arises from a multitude of publications that have demonstrated that not only the composition of a tissue, but also the localization of the cells that comprise that tissue is significant for understanding biological outcomes. For example, in a 2017 paper by Bernie Fox's lab, they demonstrated that in oral squamous cell carcinoma, the sheer number and composition of the tissue in terms of the FOXP3 T regulatory cells did not stratify patient populations. But when they looked at the spatial proximity of those T regulatory cells relative to the CD8 T killer cells, the patient population was stratified. This is just one example of how we need to be able to understand how cells are spatially related in the tissue. Now, Akoya has two platforms for multi-marker spatial analysis. There is the Phenotyping portfolio, which allows for high throughput tissue multiplexing via Opal staining and multispectral unmixing. And then there is CODEX, which is co-detection by indexing, which is a high parameter modality for multi-marker spatial analysis. And this allows for the detection of a high number of markers on cells with single cell resolution. And we'll be talking about CODEX today. So the basic chemistry underlying antigen detection using CODEX is this. We have primary antibodies that are DNA barcoded. And these primary antibodies are going to bind their target antigens inside of the tissue. We're going to reveal where these primary antibodies have bound to their target antigens using DNA barcodes that are bound to fluorophores. We call these CODEX reporters. So these CODEX reporters have the reverse DNA barcode as the DNA barcode conjugated to the antibody. And this is how we're going to be able to visualize where these antibodies have bound their target antigens inside of tissue. The workflow for CODEX is to first stain the tissue with a cocktail of DNA barcoded primary antibodies, shown here. These antibodies are going to be fixed to the tissue so that they are not removed in the following processes. We're going to reveal where these antibodies have bound to their target antigen using a one-step reveal process in each CODEX cycle, Now, this is an isothermal process by which the CODEX reporters are hybridized to the DNA barcoded antibodies. This automation for revealing where these antibodies are inside of tissue is provided by CODEX, and so we're going to reveal, image, and then remove our CODEX reporters. This CODEX cycle then can be repeated in a second CODEX cycle, in a third, so that we can collect all of our target markers in as many cycles as we need. So here I'm showing that in each CODEX cycle, we're collecting 3 targets as well as DAPI, so that we can image all of our target biomarkers using CODEX for a total of over 40 markers. So CODEX has 3 components. There's the CODEX reagents, there's the fluidics control, which automates both the fluidics and the imaging automation, and the CODEX software. So the CODEX reagents are a flexible system. We have the ability to use validated antibodies provided by Akoya, and we also have custom reagents for you, so you can conjugate DNA barcodes to your own antibodies if that's something that you would like to do. Speaking to that end, there are 3 types of CODEX antibodies. We have CODEX inventoried antibodies, and these are validated and sold as a complete assay. We have CODEX screened antibodies, which are tested for barcode and reporter compatibility. And these are not sold by Akoya, but they are user conjugated. And then we have CODEX community antibodies, which are tested and shared by CODEX users, and they're not tested or sold by Akoya. And again, the user conjugation is required. So here's an example of CODEX compatible antibodies for human fresh frozen, mouse fresh frozen, and human FFPE tissue. So CODEX is compatible with both fresh frozen and FFPE tissue, and the markers it's compatible with at this point are shown here, and we're always building in more markers to be compatible with CODEX. So CODEX also allows control of the fluidics for full automation of the system. Here we're seeing the integration of the CODEX insert with an inverted microscope. And so what we're able to do is place this CODEX insert on the microscope stage, and then we have our tissue placed on a cover slip which fits within that CODEX insert that sits on the stage, so that we're able to complete the entire imaging and revealing process with the tissue on the microscope stage. This allows for full automation of the process. So just as a summary, we're going to section our tissue onto a cover slip. We're then going to incubate our tissue with a cocktail of DNA barcoded primary antibodies. This tissue is then going to be inserted into the CODEX insert, which sits on the microscope stage. And the full integration of CODEX with the microscope can be seen here. We're then going to use CODEX to automate both the fluidics and the imaging processes to acquire 3 markers in every CODEX cycle. So here we can see CODEX automation used for stitching all of our images together for a complete 41 marker image of a 1 centimeter squared human tonsil tissue. And of course, in this particular image, we're only showing 6 markers because visualizing 41 at the same time would be rather challenging. In addition to controlling the automation, CODEX is also able to analyze the images once they've been captured. So we have 3 components to the software. There is the CODEX Instrument Manager, which is going to be responsible for controlling the fluidics as well as the microscope. There is the CODEX Analysis Manager, which is going to be responsible for data processing, including formatting the data and allowing for cell segmentation and clustering. And then there's the Multiplex Analysis Viewer, which is going to allow us to interpret our data and perform the analysis. The data that are produced by CODEX come in some simple formats. The cells are segmented, and these segmented cells in the image produce an FCS file, and we also have TIFF images. Now, also in this FCS file, we have cytometric data for each cell that's segmented, as well as that cell's XY coordinates. And this is what allows us to do spatial analysis on the cells in our image, because we have both cytometric data to determine the phenotype and then spatial analysis to determine the spatial location of that cell inside of tissue. We can create populations and identify populations of cells using a couple different strategies. One of those strategies is to use a flow gating strategy, shown here, where we can identify populations in the tissue while we gate them. The other strategy is to perform clustering, where we cluster the data and then identify cells or populations using those clusters. Once we've created our populations, we can then map them in the tissue using the Voronoi plot shown here. And we can see that with over 40 markers available to us, in this case, we're using 20 plus cellular phenotypes. I believe this particular tissue was stained with 29 markers in a tertiary lymphoid follicle of breast cancer. We can see that we have complex phenotypes in a number of them. We can have a number of them because of the multitude of markers available to us. And the Voronoi plot shown here is actually constructed based on a cell's nearest neighbors. And it's this construction of the Voronoi plot that allows us to evaluate which cells are highly interacting or weakly interacting. Those interactions, whether it's more interacting or less interacting, can be calculated and visualized in this log odds ratio shown here in this interaction matrix. So cells shown in red, or if they share a red square, that means that they're highly interacting, and the probability of seeing them interacting in this image is high. Whereas if they're shown in blue, that means that they're weakly interacting or they're less interacting. The probability of seeing those two cells together in the image is low. So in conclusion, the CODEX system enables highly multiplexed immunofluorescence for over 40 markers imaging across the whole tissue or in selected regions of interest. It allows for conversion of existing fluorescence microscope into high-dimensional cell analysis tool. It's compatible with FFPE and fresh frozen samples, and we're soon coming out with cell spread compatibility. It does not degrade the tissue, and it's compact and affordable. And now I will turn it over to Dr. Saunders.
Thanks, Dr. Carlson, and thanks to Akoya for the invitation to speak today. I work in a research lab within the Vanderbilt Diabetes Center, and I'll be sharing some of our initial studies with the CODEX system. To give you a quick preview, I'll first introduce some of the experimental goals of our research group and talk about how CODEX is well suited to help answer the questions that we're asking. Then I'll get into our experience with custom antibody conjugation and building a tissue-specific panel for studying the human pancreas. Two disclosures. One, while we are experienced with immunohistochemistry in general, this is our first time using a multiplex system. And second, we've had the CODEX system for only about 3 or 4 months, so the data that I'll be showing you today is relatively preliminary. So in the Powers and Bersova research group, we're particularly interested in understanding cellular niches during pancreatic development and also in the context of disease pathophysiology. The pancreatic islet microenvironment is composed of a lot of diverse cell types. So if you think about the pancreas as an organ, it sits below the liver in the body cavity, and it's really a bifunctional organ, so composed of 2 compartments, exocrine and endocrine. The exocrine compartment makes up about 98% of the mass, and it consists of acinar cells that secrete digestive enzymes into a branch ductal network that empties into the duodenum. And then scattered throughout that exocrine tissue are islets of Langerhans, or islets, which are small clusters of cells that are composed of endocrine cells that secrete hormones to regulate blood glucose. And islets themselves are really these kind of mini organs that are made up of alpha and delta cells, alpha, delta, and beta cells, and these are endocrine cells that secrete hormones that help regulate your blood glucose. But it's also important to remember what we consider the islet microenvironment or other cell types that come into close contact with the endocrine cells. So those include nerves as well as blood vessels, which are structurally composed of endothelial cells and also act as a conduit for immune cells such as macrophages to enter the tissue. Our lab is primarily focused on studying human tissue, and so we have developed a robust infrastructure in order to be able to answer some of these important questions about human biology. And so the way that this is set up is that we work with a number of different organ procurement organizations around the country. And when an organ becomes available for research, we communicate within our team and then actually ship that organ directly to our collaborator in Pittsburgh. And there, she's able to isolate those islets or small clusters of functional cells and also take serial sections of the tissue. So in that way, we can study both the function of islets as well as the overall tissue morphology from the same organ. And at that point, tissue and islets get shipped to Vanderbilt as well as our collaborators around the country. So this processing of the pancreas, as I mentioned, is what we've developed to be able to answer complex questions about both function and overall tissue architecture. So the primary way that we are able to do this is by taking serial sections of the tissue and processing it for different imaging modalities and other types of experiments. So for example, we have a protocol that allows us to fix tissue for electron microscopy. We also save some as fresh frozen tissue. As well as formalin-fixed paraffin embedded, and then finally a lightly fixed OCT embedded, which I'll speak to a little bit more later. And then from the regions directly adjacent to these, we're able then to isolate islets, and these are used to perform functional and metabolic studies, as well as perform some immune studies, particularly in healthy pancreata and those affected by type 1 and type 2 diabetes. So what I'll be focusing on in this presentation and showing you in some of these CODEX images are these lightly fixed tissues. And this was a protocol that was developed by Dr. Marcela Brasova, who recognized that the traditional ways of processing tissues were not adequate for the types of tissue morphology analysis that we were trying to do. So what she developed was a protocol in which the tissues are lightly fixed in a 4% paraformaldehyde solution, and then washed and equilibrated in a sucrose gradient. And this really, we found, is able to preserve the tissue architecture in a way that the flash frozen or formalin fixed tissues were not quite up to speed. As I mentioned, one area of interest for our research team is understanding how endocrine cells interact and communicate during postnatal pancreas development. So here I'll be showing you a couple of traditional immunohistochemical images where DAPI is shown in blue and endocrine cells are shown in green, red, and white. So you can see that just after birth, endocrine cells are quite dense with a lot of individual cells and just small clusters around the periphery here of the tissue. And then as the pancreas grows rapidly along with the whole body, these small clusters of cells gradually coalesce into larger islets. And overall, you can see in this last image under the childhood category, the islets are becoming less densely distributed. And this is occurring because the acinar tissue is expanding at a much more rapid pace, and so there are fewer islets shown here in kind of the same size of image frame. And if you look at the kind of zoom into the islets themselves, you'll see that the overall architecture changes during this first decade of life as well. So in the early period after birth and during neonatal and infancy stages, you see a real core mantle structure with beta cells and shown in green there in the middle of the islet structure, and then alpha and delta cells kind of surrounding in the periphery. But by childhood, as you can see by this last image, the alpha, beta, and delta cells are really fully intermingled, and this is reminiscent of what an adult islet looks like. So overall, during this first decade of life, we're seeing drastic changes in islet density as well as rearrangement of endocrine cells. And adult beta cells really have a very low proliferative capacity. And so it's thought that these early processes of both endocrine cell expansion and rearrangement may affect the future susceptibility of an individual to diabetes. And so it's particularly important that we study this time period. And it's also the case that it's during these first few years of life that autoimmunity, which is an important component of type 1 diabetes, is first observed. So for those two reasons, we're very interested in understanding more about the cellular events that are occurring. So one active area of research in our lab is looking at the context of type 1 diabetes, which has an autoimmune component. So here I'm just showing an image of an islet that's outlined in a red color, along with immune cells, CD45 positive, that are shown in brown. And so you can see by this image and the one below that there are a great deal of immune cells that are localized around the islet at the onset of type 1 diabetes. So we're interested in asking questions like which other cell types are involved in this immune infiltration, as well as the signaling molecules that are involved. So what types of cytokines or receptors are maybe mediating these events. And then we're also interested in understanding how endocrine cells interact with immune cells in a regular environment. So I'm showing here an image of a marker called NTPDase3, which modulates extracellular ATP concentrations. And it has an interesting phenotype such that it's expressed in acinar tissue right after birth. But by about 1 or 2 years of age, it becomes specifically expressed actually in beta cells. So we are wondering if this switch in phenotype may have implications for the pro-inflammatory, ATP-rich environment and how these cells might respond to that. So again, we're interested in those cellular interactions, as well as kind of generally how the islet microenvironment influences development. So I think it's clear how the types of biological questions that we're asking in our laboratory are really well suited for the CODEX system. So one of the first things that we did when we first purchased the system was to start thinking about the antibody panel that we would be building and how we would be conjugating our own custom antibodies. So this is a straightforward labeling protocol that takes only about 4 hours. It can be done on a bench top. And I just want to emphasize here that it was really helpful for us to go over the pre-conjugated list of CODEX antibodies that are available from Akoya, because that really then determines how you choose the specific barcodes that are most appropriate for the custom conjugation. And just to give you an idea of what this process is like, it first comes with all of the reagents in the kit, but first it's just a step of concentrating and priming the antibody, and then annealing to those oligo barcodes that are produced by Akoya and shipped to you. So after that a reaction occurs, you'll then just go through a few steps to purify the conjugated antibody and then run a protein gel just to verify that conjugation occurred. So here I'm showing a simple protein gel, and if you zoom in on these two lanes here that I've highlighted, the band on the left there represents a loaded antibody that is unconjugated, so you can see the weight of that antibody determines where it flows in the gel. And then on the right here, you can see that the molecular weight of that sample has now shifted due to the addition of the oligo barcode. So this type of QC just verifies that, in fact, the chemistry was successful and that the barcode is now attached to the antibody. And I've done this now for 11 different antibodies, and in my hands have had success with successfully using them in a panel in 8 of those instances. And I'll just note here that all of the antibodies that almost all of the antibodies that I've attempted to label have been monoclonal, and they've all been free of carrier proteins, which is a requirement for the labeling procedure. And once you've verified that the barcodes have in fact conjugated to your antibody, the next step is to perform a staining validation, which actually just happens on your benchtop again, not with the machine. And that's really the step where you get to assess how successful deconjugation really was. So for that procedure, the goal is really then to allow the user to specifically titrate the antibody as well as validate it in the tissue of interest that will be used ultimately in the CODEX machine. So this procedure is done in 6-well plates, just like a regular CODEX experiment. So you're actually using the cover slips that you've prepared that Dr. Carlson mentioned before, and then you're just subjecting them to the regular CODEX antibody incubation procedure. So in this case, you're incubating with the primary antibodies, or the primary antibodies that have those unique oligos. And then you'll add the CODEX reporters in this step that kind of simulates what happens in the CODEX machine during an experiment, but in this case, it'll be done on your bench top. So in most cases, the recommendation is that you use a conjugated, the newly conjugated antibody that you've just conjugated, and then you also use a conjugated antibody that has already been validated. So maybe a marker that stains the same type of cell that you're looking at and a marker that stains the different to account for kind of a positive and negative control. And then once you've done that, you'll mount your cover slip and actually visualize that stain to see and assess how your conjugated antibody has performed. Now, we ran into a little bit of trouble first thing because since we're working in pancreatic tissue, a lot of the antigens that we're interested in recognizing are not previously validated CODEX antibodies. And so we knew we were going to have to figure out how to modify this procedure in order to run positive and negative controls with antibodies that were not previously validated. And so what we came up with was, in addition to staining with our newly conjugated antibody, we also added an unlabeled antibody to that incubation step. And then before we incubated with those CODEX reporters, we actually added a traditional secondary antibody and incubated just in the storage buffer that's provided with the staining kit from Akoya. And this allowed us to successfully visualize both traditional antibody and CODEX antibody on the same slide. So that was really key in helping us to feel confident that our newly conjugated antibodies were staining the correct cells. And this slide just is a list of the markers that we've been using in our tissue samples. And I'll be showing you some images here in the next few slides, but I wanted to give you a sense of the pre-conjugated and validated antibodies that come from Akoya. Those are shown highlighted in blue. And then also the antigens that we've prepared as custom conjugated antibodies. And you can see that they represent a mix of different cell types and also obviously have a range of different corresponding reporters so that we can efficiently use multiple fluorophores in each cycle during our CODEX run. And our system here at Vanderbilt is integrated with the QIANT scope, the BCX800. The images I'll show you here are using an APO-20X lens, and the filter cubes that we are using are the DAPI GFP TRITC, which is recognizing the 550 wavelength, and then the Cy5. So this image here is a representative of one of our tissue sections that we're interested in learning more about. This sample actually comes from a donor that died very soon after birth. So this is really the kind of mid-development of a pancreas. And I mentioned before this tissue was fixed lightly in PSA prior to embedding and freezing in OCT media. And then the kind of experimental outline of this image is listed in the top right-hand corner. But we were using 8 CODEX cycles, 6 of which had antibodies involved. The other 2 were just the DAPI for registration purposes. And then we used these 18 antibodies that I showed you a couple slides back. And this particular image came from about 9 by 9 tiles at 20x. So we took 9 Z-planes, and then the CODEX software during the processing selected the best focus plane. And you can see in the bottom left-hand corner, I'm showing you different cell types that are highlighted in this particular panel. So although we used 18 antibodies, only 7 are shown in this particular image, just for purposes of color and visualizing. And so you can see that very quickly that the compartments of the pancreas that I showed you in those initial images in the introduction are very easily visualized by these CODEX antibodies, particularly the blood vessels shown in red, as well as the endocrine cells or islet clusters that are shown in that yellow, green, and blue color. And then here is just zooming in a little bit more so you can start to get a sense of the resolution from these images. And again, we've used a mixture of both the pre-validated, pre-conjugated Akoya antibodies with our antibodies. and that's really enabled us to highlight multiple different compartments of this tissue. And I wanted to give you a sense also of the range of antibodies. So this is all the same image, the same experiment, but just kind of broken down into different markers visualized. So the top left-hand corner shows the individual types of endocrine cells that we're able to look at depending on which antibody combination. The top right shows some of the vascular structure as well as some of immune cells that are highlighted by CD4 and CD89. The bottom left-hand is the image, similar to the image I showed in the previous slide, particularly highlighting the epithelium shown with the cytokeratin. And you can see some little magenta dots indicating proliferating cells through Ki67. And then the bottom right-hand panel, again, is starting to really phenotype the particular immune cells that we see around islets in the developing pancreas. So you can see we've used a combination of markers that recognize macrophages and other cell types to start to be able to phenotype these cells further. And then I wanted to give you a quick preview of the reason that we've chosen to use this lightly fixed tissue. So you'll see here these two different tissues. Unfortunately, they're not from donors of the exact same age, but kind of a similar developmental stage. So on the left is just a fresh frozen tissue, which is one of the recommended starting points for the CODEX system. And then on the right will be the lightly fixed tissue that we've kind of developed in our laboratory. So in this first row shows some of the Akoya pre-conjugated antibodies, and as you can tell from the images, they perform quite well in both the fresh frozen and the lightly fixed tissue. So you can see that this resolution is very good, isolating both individual cells as well as structures. But then when you start to look at the islet cells in particular, which we find are the most sensitive to fixation procedures, you can really appreciate, I hope, from these images that using the light fixation protocol really helps us to preserve the integrity of the cell to the cellular shape, and also to ensure that the antibodies are recognizing the antigens that they're designed to. So I just wanted to give you kind of a side-by-side comparison of our experience using those antibodies in this slightly different type of tissue. Okay, as I mentioned, we've only had this system for a couple of months, so we actually haven't had the opportunity to do a lot of the really exciting analysis yet. However, I did want to give you an example of the types of tools that can be applied to images using the built-in Multiplex Analysis Viewer, or MAV. So this is an image similar to the ones I've shown previously, and once these images have been acquired, what I've actually been able to do is to use the software in the MAV to isolate and kind of annotate the different cellular or cell subpopulations within the pancreas. So you can see in this little box in the right-hand corner a plot of individual cells within the image that are and their expression levels of two of the markers that we're interested in. So on the x-axis is chromogranin A, which marks all endocrine cells, and then C-peptide, which marks beta cells, is on the y-axis. And so I've drawn here a gate similar to the way you would draw a gate using a flow cytometer in order to specify which cells are positive for both of these markers, and then I can then assign those to a specific population. Now, you can also have the software do this automatically without any kind of user-directed input, but since these tissues are a little bit challenging to segment sometimes, we chose to do this kind of directed approach, at least for now. So once I've chosen this gate, I can essentially then apply the algorithms within the software to go through my entire image and annotate which cell types are located at each XY point. So here, the purple dots that you see are identifying non-endocrine cells, and then the yellow, blue, and pink dots are actually identifying the three of the specific endocrine cell types that we're interested in analyzing further. And then this is a Veroni plot, actually just an overlay here of the image, but you really get the sense of how intricate and how at what high resolution we're going to be able to map these specific cell types. And so some of the things that we're going to do in the future will involve applying the multi-marker analysis tools that Dr. Carlson mentioned before in order to answer some of the questions that we are asking about the interactions between cell types in the pancreas. So just in conclusion, I wanted to let everyone know that we were able to successfully integrate our CODEX microscope to be able to integrate with the CODEX software and perform image acquisition. And that also our experience with easily being able to custom conjugate our antibodies really allowed us to personalize an antibody panel that could highlight multiple pancreatic compartments. And then we did find that the CODEX antibodies that were designed for this fresh frozen tissue can actually detect antigens successfully in our lightly fixed tissue. And then, as I alluded to, we're definitely going to be using and applying the multi-marker spatial analysis tools and MAV to help us determine where endocrine cells are interacting with immune cells. Particularly, we're interested in macrophages. And then we also want to expand and adapt our panel to apply to other developmental cohorts as well as type 2 diabetes to be able to kind of get a deep phenotyping of the different cells that are present in the microenvironment. I just wanted to thank a couple members of my lab, particularly Nathaniel Hart, who did a lot of the initial characterization of the human pancreas development after birth. And also our collaborators at Stanford, Sung Kim and Yan Heng, who have been great help to us in helping to identify antibodies that are good candidates for conjugation and kind of helping getting us up and running. So I think with that, we are going to transition to our Q&A.
Okay, thank you, Dr. Saunders and Dr. Carlson, for your informative presentations. We will now start the live Q&A portion of our webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. Okay, let's get started. Dr. Carlson, let's kick off with this question. Have you seen any steric hindrance between antibodies with panels of a larger size? For example, 25 to 30 markers.
So that's a good question, and this is something that we have tested. And we have not observed steric hindrance for large antibody panels. And we also have a white paper actually for download on our website detailing how we validate our antibodies, which addresses this question.
Thank you. And, I'm going to stick with you. Dr. Carlson, you just showed a list of mouse and human antibodies compatible with CODEX. Is this the same list of antibodies that are commercially available and most up to date? And is there a shareable list of community antibodies?
So there is in that list included a subset of antibodies that are available as validated antibodies from Akoya. And we know that those lists of antibodies work with CODEX. I'm happy to share any antibody lists with customers offline.
Very good. Thank you. Let's go to our next question. How does this platform deal with and overcome autofluorescence?
So there's two ways in which we overcome autofluorescence. One way is through background subtraction. So in using the CODEX, we have the ability to hybridize and dehybridize CODEX reporters. What that enables us to do is run blank cycles, which we have no CODEX reporters in that blank cycle, only DAPI. That gives us a baseline background, which we can subtract as non-signal to go forward and remove autofluorescence. We also have the ability for FFPE tissue to use the size 7 channel instead of the FITC channel to navigate away from that high autofluorescence.
Thank you. Now, can the output of the CODEX processor be a multi-layer TIFF?
Chrissy, this is Diane. I can answer that. So we've actually been able to process our images in a way that the CODEX processor itself will stitch together an image depending on the type of scope you use And when you open that image in the image J suite, you're then able to manipulate it just like you really can with any image in image J. And so you can export it as an OME TIFF, or if you have another file format that you prefer, you can do those manipulations and export those directly from the image J with using the MAV.
Thank you, Dr. Saunders. Can the images be exported, and could they be used in other software platforms for analysis like ImageJ or similar?
So I can take this question, and the answer is simply yes. They're just TIFF images that are produced. So you can go ahead and use them with any third-party or alternative image analysis software that you'd like to use.
We have some great questions coming in. Let's see, what fluorescent channels are available, and will this be increased?
Right, so that's another good question that I can help address. So the fluorescence channels that we're going to be using are the DAPI, the 488, the ATO 550, the Sci5, and for FFPE, the Alexa Fluor 750 channels. And I don't have any information about whether or not that will be increased at this time.
Next question, is the empirical success judged by the gel electrophoresis result, actual tissue staining, or both?
So the answer to that is both. The number I showed there, the 8 out of 11, those were antibodies that have been successfully used in our panel. So I think actually 10 out of the 11 were successfully conjugated based on the protein gel, and then 2 of them, for whatever reason, did not retain their signal within the tissue afterwards. But yeah, we definitely go through the whole process before calling it a success.
Next question is, in your antibody development, did you test how long the antibody would last? In other words, how many cycles could you go without losing signal?
I'm sorry, can you repeat the question?
I can, absolutely. In your antibody development, did you test how long the antibody would last? In other words, how many cycles could you go without losing signal?
Sure, so that's a great question, and I can speak to that. So we've gone for, internally we've tested 16 cycles without seeing a loss of antigen detection. And I think a paper was recently published using CODEX where they tested 33 cycles and saw no loss of detection of the target antigen. So I can say very confidently that we do not see a loss of signal loss of signal in the ability to detect that target antigen.
Thank you. Now, I just want to quickly remind our audience, we have so many great questions coming in. Those questions that we are unable to answer today due to time restraints, and those that come in during the on-demand period, they will be answered via the email address you provided at the time of registration. Okay, let's jump to our next question. When titrating the antibody, did you formally calculate a signal, noise, or was this more qualitative?
So for the antibodies that we conjugated ourselves, it was more of a qualitative analysis. And I'm not sure if Dr. Carlson can speak about the recommended titrations for the Akoya antibodies.
Sure. So we titrate the majority of our antibodies in our control tissues for our validated antibodies. There are some subsets of tissues that we titrate specifically for those tissues of interest. And I'm happy to have a dialogue on how we do that separately after the webinar.
Thank you, Dr. Carlson and Dr. Saunders. Now our next question, Dr. Carlson, this one's for you. How do you deal with autofluorescence in FFPE tissue?
Great. So I think we might have touched on that a little bit earlier, but just to reiterate, we have 2 modalities to deal with this. There is the ability to do background subtraction, running a blank cycle, and then also using Alexa Fluor 750 instead of the 50 channel.
Dr. Saunders, let's come over to you. Were the antibodies that you used for custom CODEX conjugation already verified through IHC previously?
Yeah, that's a great question. So definitely all of the antibodies that we attempted conjugation with were antibodies that we had already screened. So we just did that on regular slides using traditional immunohistochemistry. And then once we were confident that they were working well in those tissues, then we would move over to the conjugation process.
Thanks, Dr. Saunders. Dr. Carlson, is CODEX compatible with TMA cores? Sorry.
Yeah, that's a good question. So yes, it is compatible with TMA cores, just like the rest of the tissues. You just need to section that TMA onto a cover slip.
Very good. Now, let's stick with you. What antigen retrieval is recommended for FFPE samples?
Yeah, that's a question we get quite commonly. And so this antigen retrieval is actually a lot like traditional antigen retrieval. There's some heat involved, and then either citrate and or EDTA. And the clones you're using to stain your tissue actually determine whether you're going to use citrate and or the Tris EDTA buffer.
Thank you. Dr. Carlson, let's stick with you real quick. Can CODEX data be analyzed by third-party pipelines or software packages?
Absolutely.
Excellent. Okay, Dr. Saunders, let's jump over to you. How long does it take from tissue preparation and staining to downstream data analysis for the 18 marker panel?
Yeah, so that's really going to depend on a couple different variables. The actual benchtop staining itself doesn't take very long, so it's comparable to regular immunohistochemistry. So it probably takes, including the antibody incubation, about 5 hours of prep time, and then once you get on the actual CODEX machine, it depends on how large your imaging region is. Really, is the major factor there. So I think in the images that I showed in this presentation, that I would say the average time for the experiment on the instrument was roughly 8 hours, 8 or 9, and then you're looking at another couple hours of processing, which of course can be running on a different computer while your CODEX machine is still doing something else. And we really haven't done a large majority of analysis yet, so I can't really speak to that. I think it kind of depends on how granular you want to get, but yeah, I would say from start to finish, probably you're looking at least 12 to 15 hours of kind of getting the data out.
Thank you, Dr. Saunders. Now, let's stick with you. How do you store all the data generated from each run? And as you scale up CODEX experiments, how do you plan to address data storage?
Yeah, this is kind of a source of concern for us just because we are planning to screen a large volume of tissues. So we have some network-associated storage available at our institution that we're taking advantage of, as well as some cloud-based backup and some other servers available. So I think it definitely is a situation where you want to have a conversation with the IT department or the infrastructure within your institution or company to make sure that you have kind of the bandwidth to be able to store these large image files, sometimes up to a gigabyte for just a single experiment. So I think in the same way that sequencing data requires a lot of storage space and of kind of forethought and planning when you implement a new system, you just kind of have to be ready for that influx of data.
Thank you, Dr. Saunders. Now, here's a question. How large are these images and what kind of IT support is necessary?
Yeah, that's great. So I'll take that question. So the size of the image is going to be dependent on really the size of the tissue. So it's hard to give a firm number for that, but we're on the scale of like gigabytes of data here, not megabytes. And in terms of the IT support, we help you with the installation and setup of everything. Whether you want to set up some servers or have an alternative setup beyond what our standard installation is, that's totally up to you. We're flexible, and it really depends on what you want to do.
Thank you. Can I use flow software to analyze output data?
Yeah, that's also a good question. So the two types of files that are produced by the CODEX are the FCS files and the TIFF images. And so flow data, or the flow software, is going to be able to process those FCS files. So yes.
Okay. Thank you. And this is for Dr. Saunders. what was your evaluation of the efficiency of CODEX's cell segmentation pipeline? Did you try an alternative method such as cell profiler?
Yeah, that's a good question. And for those of you who don't study the pancreas, it's kind of a difficult organ to work with in some cases. So cell segmentation is a little bit more challenging than some other tissues. So we didn't evaluate really another pathway side by side. We typically in our lab use Indica software to do our analysis, our algorithm-assisted analysis. So, you know, comparing it to the Indica Labs software that I've used, I would say that it's just as good, if not better. But I haven't gone through in a detailed fashion and compared it to other systems.
Thank you. What is the optimal tissue thickness for CODEX analysis?
So I can speak a little bit to this. So the tissue has to be between 5 and 10 microns thick, so it needs to be somewhere in that range.
And I think the tissue, all of the tissues that I showed you were between 8 and 10 microns. So, you know, depending on how large the section that you're trying to image is, or sorry, how large the area is that you're trying to image, you will sometimes need to adjust the range of Z-stack. So when, like I mentioned, the processor during the analysis, pre-analysis during the processing phase, will select the best plane essentially for each tile. And so the thickness of your section there might mean you have to take more or less Z tiles in order to get that best focal plane.
Thank you. Now, can you do multi-point imaging of just islets and exclude exocrine tissue?
So I think this may be a question about like regions of interest, and that is, Grady may be able to answer this a little bit better than me, but you can set multiple areas of analysis during different or during the same imaging run. So you wouldn't necessarily have to take that full section image like the one I showed. You could choose individual locations within it to image.
Yeah, I think you've captured that well. You could choose to image those regions or just do the analysis on those regions afterward, whichever way you want to manage the data.
Thank you. Can you do multi-point, oops, so sorry. I was reading the same one. We have time for one final question, and let's wrap with this one. And Dr. Saunders, I believe this one's for you. Do you see the need to go up to a 40x objective in the future?
Yeah, that's an interesting thing to think about for us. We've traditionally stuck at 20x and feel that that's an adequate kind of compromise between really being able to capture the multicellular nature of the islets, but also to be able to really easily capture the kind of spatial organization of the cross sections of the tissue that we're interested in. So I would say that the majority of our work is kind of looking at cellular interactions and not so much at focusing in on individual cells. So we don't have any plans in the near future to go up to 40x, but I think certainly it would be, it would produce beautiful images. So maybe someday we would consider that.
Okay, and you know what, we're going to fit in one quick question. And Dr. Saunders, this is for you. When validating custom antibodies in the pancreas with a primary-secondary approach, did you use a clone targeting a different epitope?
Oh, that's a great question. Yeah, so in most cases, I actually used a completely different antigen, but an antigen that stained for the same cell, if that makes sense. So you definitely could use the same, an antibody that was targeting a different epitope of the same antigen, but in our case, we actually just completely switched, but in a population that we still knew should be positive for the same markers.
Thank you. Now I want to thank Dr. Saunders and Dr. Carlson for their time today and for their important research, and we've had an excellent Q&A session. And I'd like to also extend that thanks out to our audience for joining us and for their interesting questions. Again, questions we did not have time for today. There were some that came in we don't have time to answer, and those that are submitted during our on-demand period will be addressed by Dr. Saunders and Dr. Carlson via the email address you provided at the time of registration. Now, before we go, we'd like to thank LabRoots and our sponsor, Akoya Biosciences, for underwriting today's educational webcast. Now, a poll will pop up on your screen, and your participation is greatly appreciated. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thanks for joining us today, and we hope to see you again soon. Have a great day.